So what is this lac operon? It is also a kind of regulatory system, but that is seen in the bacteria cells, okay, especially in this E. coli. So remember, it is a regulatory system that is seen in the bacteria E. coli. Okay, so what does it will do? Regulatory system. We know that it will control the expression of genes, right? That involved in the lactose metabolism. Remember this. So what does the lacoperon will do? It is a regulatory system that will control the metabolism of the lactose. Same like that. So we know the E. coli, it mainly prefers the glucose. So most of the time, E. coli doesn't want the lactose at all. It only prefers the glucose. But if the E. coli doesn't have the glucose, then it has to depend over the lactose. So during that time, we need these proteins. So these are a kind of luxury proteins we need only at a particular point. Understood? So you can say the lac operon, these are non-constitutive proteins. They required only at a certain time. When the E. coli doesn't get the glucose, it depends on the lactose. So for this lactose metabolism, we need these proteins. Understood? So see here, however, sometimes the absence of glucose, lactose has to be utilized by E. coli as a substitute for energy. Okay, so lactose or beta galactidase, you can also call the lactose as beta galactidase, which is a disaccharide. Okay, it will be containing dimeric sugar that we'll be studying in the biomolecules. So just understand that lactose, it is a disaccharide. That means there are two sugar molecules are present. One is the glucose and the galactose. Understand this. So lactose is equals to glucose plus galactose. If you break down this lactose, you will be getting the glucose and the galactose. Understood? Is that clear?
Yes.
Okay. So if you see the structure of this lacoperon, it consists of mainly three genes. One is the LAC-Z, LAC-Y, LAC-A. Okay. So remember this. There is a LAC-Z, LAC-Y, and LAC-A. These are the gene areas. These are the structural gene areas that is present between the operator and the terminator region. Okay, so before this operator region, we will be having the promoter region for the structural gene. Before that, we will be having another gene area that is called the LAC-I. So what does the LAC-I will produce? It will produce the repressor. Understand this? This is a LAC-I is a LAC inhibitory gene which will produce the repressor. So that repressor will go and bind to the operator region so that the RNA polymerase cannot synthesize the LAC-Z, LAC-Y, LAC-A genes. Understood? So remember, there is a PI, LAC-I, PO, LAC-Z, LAC-Y, LAC-A, and terminator. So this is the LAC operon. Okay, so in this LAC-Z will produce the beta galactidase enzyme. Remember that. So what does it do? It will produce the beta galactidase enzyme. Okay, and LAC-Y, it will produce the permease enzyme. I'll explain you what are all these enzymes will do. And LAC-A, it will produce the trans-acetylase enzyme. Okay, so remember there is a lactazine which codes for the beta galactidase enzyme. This enzyme, it will be helpful for breaking down the lactose into glucose and galactose. Remember this. So whatever the lactose is there, it will break down into glucose plus galactose. Understand? And what does this permease enzyme will do? It will make the cell membrane permeable. Okay, so permease, it is a membrane bound enzyme that makes the E. coli cell permeable for the lactose. Okay, remember this. Membrane bound enzyme. It will make the cell membrane permeable. What is meaning of permeable? It is like a porous structure where through these pores the lactose can enter. Okay, so if it is more permeable, it can uptake the more lactose. Usually bacteria doesn't want the lactose. It depends on the glucose. But when you don't have the glucose, now the bacteria has to take out the lactose for its energy source. So this permeable enzyme, this permease enzyme, it will make the cell membrane permeable so that the lactose will come inside more freely. So more and more lactose can come inside and the E. coli can use this lactose for its energy source. Understood?
Sir, I didn't understand the permease part. So can
Okay,
I
see here.
come?
Yes, I'll explain them in detail. Okay, don't worry. I'm just giving the rough thing. So just understand that permease enzyme, it is permeable so that the lactose will
So,
come inside the
I'm
cell.
audible.
Yes, Banu. Banu, tell me.
So, I'm rejoining, sir. The whole video was cutting till now.
Banu, your
Last.
net is very bad. Even your voice is breaking down.
So, I'm audible.
Yes, Banu, you are audible.
Sir,
Banu, you are audible. You are audible.
Sir, she says
one
you're.
second, I'm joining.
So this lac operon, it contains the inhibitory gene. So what do we call this lac as? It is a inhibitory gene. Okay. So what does that inhibitory gene will do? It will not allow the lac enzymes to be produced. Okay. So whatever the polymerase enzyme that is attached here, it has to pass through this operator region. But before passing through this operator region, when it passes through the lac, it will produce the repressor. That repressor, now it will come and bind to the operator region. When it bind to the operator region, this RNA polymerase can come up to here, but it will be facing the difficulty to pass through this region because there is obstruction by the repressor present here. Okay, so this enzyme will be not produced, this enzyme will be not produced, this enzyme will not be produced. Understood? Is it making sense? What happening here? So remember, this is a inhibitory gene.
Yes, sir.
That inhibitory gene will produce the
So, can you
repressor.
send for this recording, sir?
Yes, yes, Banu, I'll send that. Okay. Anyways, we just discussed only few things. That's okay. Listen from here carefully. So what is this PI? This is the promoter inhibitory gene for this LACI. Remember this. This is promoter inhibitory gene. Promoter for this inhibitory gene. So you can say it is a inhibitory promoter for inhibitory gene. So this is the promoter for inhibitory gene. This is the gene inhibitor. This is the promoter for the structural gene. So promoter for actually the lag gene, you can say. Okay, promoter for lag gene. So that's a lag Z, lag Y, lag A. And then you will be having the terminator. Before reaching this, you will be having the operator where the repressor will come and bind. Understand? So see here, RNA polymerase binds to the promoter of the inhibitory gene and it will pass through the LAC, which is the gene inhibitory region that will transcribe the repressor. Okay, that will produce the mRNA which will code for the repressor. So that's what it says. It transcribes the repressor mRNA. So from that repressor mRNA, you will be getting the repressor protein after the translation. Is it clear? Mridula and Banu, is
Yes,
this
sir.
point clear? The first point.
Sir, the RNA polymerase will bind to LAC-I gene, and
Hmm.
after that,
No, no, Banu. Sorry, no, Mridula. First, what
yes.
will be having? We'll be having promoter region. To that
Mm.
promoter region, RNA polymerase will bind. Yes or no?
Ah.
So this promoter is for what? For a particular structural gene. Yes or no?
Mm. Mm.
This promoter is for this gene. This promoter is this for this gene.
Okay, okay.
Understood? So this is the
Mm.
promoter for this gene.
Mm.
So that's why it is called as PI because promoter for inhibitory gene. And
I
what is
gene.
LACI? LACI is nothing but the inhibitory gene. Remember, this is inhibitory gene. Why we call this as inhibitory gene? Because it will produce the mRNA codes for the repressor. And that repressor will produce the protein repressor. So this repressor will bind to the operator region. Understood?
Yeah, sir, understood.
Okay. So remember, PI is it a inhibitory gene promoter, whereas this P is a promoter for LAC-Z, LAC-Y, and LAC-A. This is a polycistronic. You can say the single mRNA, I mean single gene is coding for 3 different proteins. So you can call it as polycistronic. Understood? What is monocistronic? That means you will be having only one gene between the operator and terminator. For suppose operator and you have only the lag-Z and then terminator, then you can call it as monocistronic because it's only coding for one gene. Whereas if it is 3 types of proteins are coming from this, then you call it as polycistronic. Understood? What is meaning of this?
Yes, sir.
So just listen. First, we'll be having promoter inhibitor gene that codes for LAC-I. Okay. So the RNA polymerase will come and bind to the promoter inhibitor gene. There you don't have any operator region here in between. So it simply go to the LAC-I gene area. It will synthesize the LAC-I mRNA. What is that called? The repressor mRNA. And this repressor mRNA will undergo translation process and it will produce the active repressor. Now, instead of having the operator region, generally what happens? We will be having promoter, then the laxine and the operator region, so that the transcription will end. But it will not be ended here. It will continue to the another gene, that is the promoter region. Another promoter region. That promoter region, it codes for laxine, LAC-Y, and LAC-A. So this after this promoter region, you will be having the operator region before you enter to the structural gene. So whatever the repressor that is produced here, this repressor will go and bind to the operator region. So once this operator region is bound by the repressor, then the polymerase enzyme will stuck at the promoter region. It cannot move forward. So no transcription will happen for LAC-Z, LAC-Y, LAC-A. Understood?
Yes, sir.
Banu, what about you? Is she there in the class?
She is there.
Okay. So next one. So repressor mRNA, it is translated into the active repressor protein. Okay. So whatever the repressor mRNA is there, it will produce the active repressor. Now this active repressor, it has the affinity for the operator region. So it will go and bind to this operator region. Okay. So after that, the next enzyme will come and next enzyme will also bind when this was leave off. Okay, see, that's what he says. Another repressor protein is already translated and formed, that which is sitting here. But this will be expired, so the new one will come and bind it. Okay, so since repressor is present at the operator, RNA polymerase that cannot move forward and it falls off. Understood? So whatever the RNA polymerase that has reached to this promoter, it cannot go further because the repressor has bound to operator region. So the gates were closed. As the gates were closed, the RNA polymerase will just stay here and fall off. Understood, Mridula?
Yes, sir, I understand.
So that's what PI means, promoter for inhibitory gene.
Stop.
LAC-I means inhibitory gene. Regulatory gene for LAC operon expressed constitutively codes for repressor protein. So remember this. This is expressed constitutively. Which one? The LAC-I gene. Whereas if you see this one, this is non-constitutive. And this one is a constitutive. Understood,
I'm
Mridula?
audible.
Yes, Banu, you are audible now. What happened? We
That
have
is
a,
the whole class is now again.
what happened?
So I have a network issue, sorry.
Banu, you don't have a Wi-Fi.
The Wi-Fi only was not working, so now I'm using mobile data, but still your voice is breaking. So
Okay, what shall we do? So just connect to a proper internet. I
just
can
send
give
me
you
the
time
video so
if you
I'll
want.
rewatch it again.
Yeah, that's okay. But we just discussed only 10 minutes. What about the things that we discussed? Please watch it carefully.
Yes, sir.
Yeah. So see, remember this is a constitutive gene because it doesn't have any operator here. Yes or no? It is not regulated. Whereas you see here, this is a non-constitutive gene because it is regulated by operator. Ridula, you understood this?
Yes, sir, I understand.
Yeah, notice these points. Okay, so one thing, PI, it is for LAC-I. P for laxins. Operator region is present between the P and the laxins, whereas no operator region present between the PI and LAC-I. So this is a non-constitutive. This is a constitutive gene. That means they are produced constitutively, housekeeping genes. Whereas this one, it is a non-constitutive genes. These are luxury genes, can be produced only at a particular situation. Understood. And this LAC-I, it will produce the repressor mRNA that will produce the active repressor, which is a protein that will bind to the operator. So this is the inhibitory protein. Remember this. This is also called as the auxiliary protein, or you can say this is the inhibitory protein or regulatory protein. Understood this?
Yes, sir.
Okay, so you can see here
Yes, sir.
PI, LAC I, PO, LAC Z, LAC Y, LAC A, terminator. So P for promoter of lacsin, O is for operator for the lacsin, where the repressor protein synthesized by LAC A will come and bind here. And lag genes, these are the lag Z, lag Y, lag A, which is the actual structural gene. Okay, and this is a polycystonic because if you see between the operator and terminator, you are having three different genes. Usually, between the operator and terminator, if you are having only one, then you call it as monocystonic. Between the operator and terminator, if you are having three or more than one, you call it as the polycystonic. And remember, these enzymes, whatever that are produced, they will be helpful for lactose metabolism. Okay, whether they will convert the lactose to glucose or whatever the lactose, it will produce some toxic elements. To in order to metabolize those toxic elements, we need the enzymes that will deal in detail. Understood this?
Yes, sir.
So just give me a 1 minute. Meanwhile, you guys just read this thing, okay, and just note. Okay, see here. First one is the laxizine. Remember the order also. Order is very important. The first one is laxi, and then you will have Y, and then you will be having A. Okay. So what does the laxi enzyme laxizine will do? It will produce an enzyme that's called the beta galactidase enzyme. Remember this. Okay. So which enzyme it will be produced? It is beta galactidase enzyme. Okay. So what is the function of this beta galactidase enzyme? It will cleavage the lactose into the glucose and the galactose. It will break down the lactose into glucose plus galactose. Remember this. Okay. So it is a very, very crucial for the lactose metabolism where it will be breaking down the lactose into glucose and the galactose. Okay. You can also call this enzyme as hydrolyze enzyme. Remember this. Sometimes they can also say like this. It is a hydrolyze enzyme. Why we say hydrolyze? Because it is hydrolyzing the lactose into glucose and galactose. Hydrolyze means you break down into smaller units. So is it a catabolic or anabolic reaction? You know what is catabolic and anabolic reactions in the metabolism?
Is it
Remember?
metabolic,
Yes.
sir?
Hmm? So remember, catabolism means whatever the bigger molecule is there, you are breaking down into small pieces. This is called catabolic reaction. Anabolism means whatever the small pieces are there, you will join them into and make into a single bigger molecule. That's called anabolism. This is catabolism, this is anabolism. So this comes under catabolic reaction, where you are breaking down the bigger molecule into the smaller molecule, glucose and the galactose. Understood? So you can also say the disaccharide, lactose is a disaccharide. The disaccharide is converted into the monosaccharide, two monosaccharides. Okay, is that clear?
Yes, sir.
Yeah.
Yes, sir.
So next thing, you will be having the lactazine. So lactazine, it will be coding for the permease enzyme. Remember this. This is called the permease, or you can also call it as the lactose permease. Okay, so this is also said to be the lactose permease enzyme. Okay, so what is the function of this? It will facilitate the lactose transport into the cell. Okay, for suppose you have a cell like this. So this cell is having the pores through which something can go from outside to inside the cell. You can say they are like a tunnels from which the lactose can pass through the cell membrane. Okay, so through this tunnel, the lactose will go from out of the cell to the inside the cell. So this permease enzyme will allow this to go. Okay, what are these tunnels? These tunnels are nothing but the permease enzyme. If you have these tunnels, then only the lactose can pass through the cell membrane. If the tunnels are not there, and if it is a continuous cell membrane, then the lactose cannot pass through it. Understood? So these enzymes will go and bind to the cell membrane and makes the tunnels. Those tunnels will help the lactose uptake. Understood?
Yes, sir.
Yes,
Yeah. So remember this. Okay. So it will pump the beta galactidase into the cell. And remember, this is a protein gradient. That means it requires certain energy to take that. Okay, that will not deal. That's not needed. Next one, the LAC A enzyme. Okay, so this LAC Z, sorry, the LAC A gene, it will code for the transacetylase enzyme. Okay, so what does this transacetylase enzyme will do? It will it will transfer. Remember, I'm not keeping the ink mark, so just notice somewhere if there is any additional points I'm adding. So remember, it will transfer the acetyl group from acetyl coenzyme A to beta galactidase. Okay, this is not needed, but just remember that's it. Okay, just know it. So it will transfer the acetyl group that is transferred from acetyl CoA to beta galactidase. Just remember that. Beta galactid, sorry, this is not ACE, this is a beta galactidosides. There's some other chemical. Just understand that it will transfer the acetyl group from acetyl CoA to beta galactidases. Okay, understand that. So why does this transfer will happen? So this transfer, it plays a very important role for detoxification. Okay, so actually there will be certain things are produced, like non-metabolizable thioglycosidases are produced, which are very toxic material. So in order to detoxify that, we need these lacasins. Okay, understood? So they will do the trans-acetylation reaction, that is transferring the acetyl group from one compound to other compound, so that neutralizing the detoxificants. Any doubts so far? You understood everything?
Understood, sir.
Yes, sir.
So what is the function of laxizine? Tell me. Bonnie, you tell me, what is the function of laxizine?
Sir, codes for beta galactosidase enzyme.
Mm-hmm. So what does this beta galactidase enzyme will do?
Sir,
It will break down the lactose to glucose and galactose.
okay,
Remember that. Okay.
Yes, yes.
One more thing I forgot to add here. Just remember, it's not only the lactose. There is a similar kind of disaccharide that we call it as yellow lactose, which is just similar to lactose, but it's a somewhat different yellow lactose. Okay. So this yellow lactose also be converted into the glucose and galactose with the help of beta galactidase enzyme. Remember this. Okay, this is very important. Please just note down this one. Okay, yellow lactose or lactose, they both will be converted into the glucose and galactose. Okay, so this
So
is a
what is the
disaccharide. This disaccharide will break down and convert into monosaccharides. Clear? And
Yes.
remember, this yellow lactose and lactose, they acts like an inducer. Remember this. This is also add one thing. I'll explain this again, but they act like an inducer. So you know what is meaning of inducer? They will induce something. So what does do? They are like inducer. What they will do? They will attach to the lag repressor, and whatever the lag repressor which is activated, they will make them inactivated. Understood? So what is this lactose and lactose will do? It will be acting like inducer. That means they will attach to the repressor. When it is attached to the repressor, what happens? What happens when it is attached to repressor? It will, whatever the active repressor is there, it will become inactive. So now this inactive repressor cannot bind to the operator region. Okay, so there is no repressor binding to operator region. Now what happens? The RNA polymerase can easily pass through the operator region, and it reads the LAC-Z, LAC-Y, LAC-A and synthesize the enzymes. So lactose itself will regulate the process. So the bacteria doesn't have the lactose. I mean, it is having glucose. So just remember like this, like a story. For suppose, let me just remember this like a story. So what happening here? So initially there is a bacteria cell. So there is a bacteria cell. This bacteria cell will depend on the glucose. So the cell always takes the glucose. Okay. So it is having enough amount of glucose, so it doesn't need to depend on the lactose. But what happened now? This cell, it is lost the glucose. So it is not finding any glucose. So the glucose is the primary source, but you don't have this primary source. Now the bacteria, it has to depend on the lactose for its survival. Okay. So what happens if the cell is having all the glucose? Okay. So in this condition, what happens now? So there is a promoter I gene, and there is a lac I gene, and then promoter, and then operator, and then lag genes. Okay, this is like a lag gene. Remember this. So what happens now? When the glucose it is taking, it is depending on the glucose. So the polymerase enzyme will come here attached to PI and it will pass through LACI. This LACI will produce the repressor. This repressor is active repressor and there is no one to suppress this active repressor. Now this active repressor will go and bind to the operator region. So the enzyme will come here, promoter region, but it cannot move forward. Why? Because the repressor is attached. So this will fall off and the transcription ends. But if the glucose is not there and lactose is there, now this lactose or A-lactose will convert this active repressor to inactive repressor. So now this inactive repressor, it cannot bind to the operator region. So this enzyme can pass through and it will read the LAC-Y and it will come to the ending terminator region. And it will synthesize all the enzymes that are required. Beta-galactidase, trans-acetylase, and permease enzymes. Those which are required for lactose metabolism. So the lactose is itself the inducer for this active repressor. So that's why it is act like a luxury. Only when you are depending on the lactose, then only this enzyme will be inhibited so that the genes can be synthesized. But you don't have the lac, then all the times it is not synthesizing the lac genes. Understood this? Is it making any sense?
Yes, sir, understood.
What about you, Banu?
Yes, so can you explain again?
Hmm? Banu, please tell me again. I didn't get you.
So
Banu,
if
am
you
I?
can explain again, I'm
Okay, just to understand this. There are two
yes,
scenarios
sir,
you take.
yeah.
Scenarios you take. Scenario 1, scenario 2. Okay, scenario 1, when bacteria depend on glucose. Scenario 2, glucose is not available. Okay, so glucose is absent. Now bacteria depends on lactose. Okay, so when bacteria depends on the glucose, what happens? The repressor, active repressor, it will always remain active and it will bind to the operator region. It will bind to the operator. Whereas here, what happens when lactose is present? So this active repressor, it will become inactive repressor. How it become inactive repressor? Because there is a lactose will bind to it. Okay, so whatever the repressor is there, for this repressor, the lactose is bound and make this repressor inactive. Now this inactive repressor cannot bind to the operator. When it cannot bind to the operator, transcription happens. So transcription takes place for laxine. Whereas here, when it is bound to the operator, the RNA polymerase cannot move forward, so transcription doesn't take place. So transcription doesn't take place. Understood these two scenarios?
Yes, sir.
Okay, Banu. Banu, once just go through the PDF. If you have any doubts, I'll take you a separate class and I'll clarify all the doubts, okay?
Yeah, yes, okay.
I mean,
Yes.
you just missed the beginning of the class, so okay, fine.
Yeah, okay.
So understand these things. Bridra, till here, no doubts, right?
No, sir, I understood.
Okay. Next one. So lac operon, it was given by these scientists, the Francois Jacob and Jacques Monod, who is biochemist and genetist. Okay. So operon is defined as a polycistronic gene, okay, regulated by common promoter regulatory protein. Okay. So remember this. It is having a common promoter and the regulatory protein. If you see here, just see here. If you see, this is a one gene, gene 1, gene 2, gene 3. All these genes, they are having common promoter and common operator. Okay, so that's why it is a polycistronic. Understood? What is this point says? So usually wherever if you see the operon word, operon means polycistronic. Remember this. So you have a lac operon like that. There are other operons also there in the bacteria cells, like TRP operon, ERA operon, IS operon, VAL operon. Understood? So all these operons are nothing but the polycistronic genes regulated by a regulatory protein, that is repressor. So here it is a repressor. Understood?
Yes, sir.
Okay. So both of them got the Nobel Prize. Know this. Now you tell me. So what happens when the E. coli doesn't feed on lactose?
The transcription will be stopped.
Yes. So what happens here when the E. coli doesn't feed on lactose? The lag gene will be off. There is no transcription. There will be no synthesis of the proteins. Okay. You can E. coli doesn't feed on lactose. This only happens when the E. coli is having enough amount of glucose and it doesn't need on the lactose. Understand? So read here. E. coli doesn't feed on lactose normally because E. coli prefers the glucose over the lactose. Remember this point. E. coli prefers glucose over lactose. And this RNA polymerase, it binds to the promoter region of the inhibitor gene and it will pass through the laci and it will transcribe the active repressor that will bind to the operator region. So whatever the repressor is found, it will go and attach to the operator region and the transcription doesn't take place. Hence the lag gene expression is off. Understood? So next one. So next one is E. coli feeds on lactose. When does this will happen? When the E. coli, it encounters the environment where the lactose is only present but not the glucose. Understood? So there is no glucose, only lactose is present. You can see here the orange one are of glucose, whereas here there is no orange ones, only the kind of light yellow ones are present. Okay, so they're all lactose. So now the small amount of lactose will enter into the cell, okay, through the help of permease protein. I told you what is permease protein. It will be like a tunnels which will pass through the cell membrane. It will deliver the lactose from outside the cell to inside the cell. Inside the cell, once it reaches, whatever the lactose are there, they will be converted into glucose. Yes or no? Or you can say this lactose, it will be converted into the A-lactose also sometimes. It is like a modified structure of this lactose. So it will be converted into yellow lactose. Now this yellow lactose can be converted into the glucose and all. Okay. And remember this yellow lactose or lactose, they will also bind to the active repressor. So when it bind to the active repressor, now the LACY were synthesized. So now this LACY is also permeable. So there is more and more tunnels were added, more and more lactose will come inside the cell. Understood?
Yes, sir.
So it will
Yes, sir.
transcribe the LACZ, LACY, LACA so that the lag gene expression is on. Okay, so RNA polymerase bind to the promoter, inhibitory gene, transcribes the repressor mRNA and forms the active repressor. This active repressor, the lactose will bind. This lactose will make the active repressor to inactive repressor. So this inactive repressor cannot bind to operator. So the RNA polymerase can happily pass through the operator region and synthesize all these enzymes. So here the lag gene expression is on. Understood? And remember this permease, what it will do? It will bind to the cell membrane. When it bound to the cell membrane, what happens? There is more tunnels. When there is a more tunnels, what happens? There is a more incoming of lactose. Okay, when there is a more incoming of lactose, what happened? Now whatever the effect is there, it will be amplified. Remember this thing, it is not given anywhere, but just know this. Why it is amplified? Because the permease is attached to the cell membranes, more lactose are incoming. Because of that, more active repressors will convert into inactive repressors. So more lactose metabolism will happen. So it is amplifying because of permease. Sometimes they can also give you like questions. So which of the enzyme will amplify the lactose metabolism? It is a permease enzyme. Understood?
Yes, sir.
Yes, sir.
And remember, this lactose is an alternative energy source for the E. coli. The primary source is glucose. When the glucose is unavailable, it will depend on this lactose. So that's what the scenario 2. So just go through this, whatever that we discussed, the same thing. So this is what happened. So lactose absent, repressor protein will be active, regulation will be negative, switch off. Lactose present, inactive, no regulation, switch on, transcription happens. So gene expression, or you can say it is a transcription plus translation. Okay, if you confuse. So transcription happen and then translation will happen. Clear? So lactose, its isomeric structure is also called, I told you, the lactose, okay, which is also inducer and inactivates the repressor binding. Remember, if the option is given with both lactose and lactose, select the lactose. Okay, so this lactose has to be modified to lactose, then only it can bind to the inducer. I mean, it acts like inducer, we will bind to the repressor. Understood?
Yes, sir.
Yes.
What is this? See, the same way, if you are having the repressor who will stop the process, there is also sometimes you will be having the activator who will speeden up the reaction. Okay, so remember, activator, generally if it is happening in normal phase, because of this activator, the pace will go so high. I'm saying about the pace, the speed will go high. So those are positive regulation. So see here, normally RNA polymerase binds to DNA, scans the promoter region in the entire DNA. Okay, so positive regulation, call the activator present in the cell. This will bind to the RNA polymerase, helps it to reach the promoter region fast. So it will not delay it, because we know the promoter region has to find out where is the RNA polymerase, it has to find out where is the promoter region is present. Okay, so that's a Tata box. It has to find out where it is. So this one will quickly locate where the promoter is there, and it will tell the RNA polymerase, please come fast here. That's what it do. So now this will come quickly without wasting any time. So the gene expression will be fasten up. Understood? So see here, so it reaches to the promoter region faster. So the transcription initiation will increase. So positive regulation will increase the gene expression. Understood this? So you see here, no regulation, positive regulation, activator, negative regulation, repressor. Positive regulation, activator will be associated with the RNA polymerase enzyme. Whereas repressor, it is associated with the operator region. Remember this thing as well. Okay, so one will be fast in the reaction, the other one is small in the, not reaction, fast in the transcription, the other one will be decreasing the transcription. Clear? So tell me,
Yes.
what is the answer for this? This is a trans-acetylase. Remember, this is a trans-acetylase.
Sir, permease.
Which
And
enzyme will be produced in a cell if there is a nonsense mutation in the LAC-Y gene? LAC-Y is producing for?
permease.
Yes. But what is saying here, producer in a cell? Which enzyme will be produced in a cell if there is a nonsense mutation? Read it carefully. Read it carefully. That's what I'm thinking. Yeah, read it carefully. Understood? What is the answer now? The answer is D, beta galactidase. So see here, what he's saying, which enzyme will be produced in a cell if there is a nonsense mutation in the LAC-Y gene? So go back. So what he's saying, there is some mutation which happened in the LAC-Y gene, and it will not allowing the LAC-Y to express. So it is not expressed. So if it is not expressed, then it is the terminator. So this will also be not expressed. Then which of the enzyme will only be produced? Only the LAC-Z. That is nothing but the beta galactidase. Understood? Read the question carefully. This is what happens. You may think that this is the answer, correct answer, but no, please read carefully the question. Understood this?
Yes, sir, got it.
Yeah. Just
Yes,
be
sir.
careful in the exam. Whenever you are solving the question, don't hurry up. Read carefully. So that's what happened. Beta galactidase will be produced, but the LAC-Y, LAC-A will not be produced. Understood? So next one, eukaryotes regulation. Eukaryotes regulation, I think I already explained now. I told you where does the regulation will happen. Either it will happen on the transcription process or it could happen in the some other process. Do you remember?
Yes, sir. Yes, yes, sir.
Ritula, what about you?
Yeah, sir. Remember.
We'll do one thing. You have a class now, Ritula?
Yeah, sir. I told at 4:30 I have class.
Okay. Then do one thing. I'll be sending this entire PDF. I think already sent no. So just go through it. If you have any doubt, then I'll explain it. If you understood, then I'll not explain. We'll move to the next topic. Is that clear? Stage 1, stage 2, stage 3. Just I gave you a rough explanation. Just understand that. I told you now where the transport of mRNA will not happen or translation will not happen. So just go through it. Is that okay?
Yes, sir. I'll go through.
Okay, guys, then. Bye bye. Take care.